Hello dear students, welcome to biology class. So today we are moving to a new chapter in second PUC, that is human reproduction. So let me explain the very important topics covered under this particular chapter. Then we will move to the MCQs on this particular chapter. So now let us study the important points. So in this particular chapter we are mainly focusing on the structure of reproductive system as well as the process of production of gametes, that is called as gametogenesis, followed by the fertilization events as well as the post-fertilization events that occurs during the human reproduction. So at the beginning, let us study about the reproductive events in the humans in general, just an outline of the reproductive events that occurs in human beings. The first event in human reproduction, the very important event, that is gametogenesis. And the gametogenesis is nothing but the formation of gametes followed by the insemination. Insemination is the step responsible for the gamete transfer. The transfer of sperms into the female genital tract is called as insemination. Insemination is followed by the contact between the male and female gametes. So that leads to the fertilization. So fertilization is nothing but the fusion of male and female gametes, followed by cleavage. So this is the development, the formation and the development of blastocyst, and then implantation, nothing but the attachment of a blastocyst to the uterine wall, followed by the gestation. So gestation is the period of embryonic development and followed by the parturition, the last step in the reproductive process. Parturition, nothing but the birth of the child, the delivery of the baby. And one more event, that is lactation. So after the childbirth, the mother will provide the nutrition to the child in the form of breast milk. So that is the production of milk. So these are the very important events of human reproduction. And as we all know, with respect to the reproductive system of human body, or in case of human body, the structural components are very different and the events involved in the human reproduction are very complicated. If you consider all the other living organisms, the reproduction in human beings is very complicated. And you know, in case of human beings, there is a prolonged period of parental care, 9 months, 7 days of gestation period, followed by the longer duration of parental care. So with respect to the structure as well as with respect to the events that occurs during the reproduction, the human reproduction is very complicated. So in this chapter, we are focusing on all the events that occurs during the human reproduction. Now, before entering into the mechanism or the structure of human reproduction, let us see the important differences between the male individuals and the female individuals. So what are the important differences between male and female individuals with respect to the reproduction? There is a difference in the structure of reproductive system, definitely. And even there is a difference in the gametogenesis process. That means the type of gametes produced. You know, the human beings are the unisexual individuals. The male individual produces male gametes and the female individual produces female gametes. And the sperm formation continues even in old men, but formation of ovum ceases in women around the age of 50 years. So this is another very important difference. The duration of reproductive phase, it shows great difference between the male and female individual in human beings. So these are some of the very important differences between the male individuals and the female individuals with respect to the reproduction. Now, let us focus on the structure of male reproductive system. So what are the structural components and what are the important functions of each structure involved or found in the reproductive system? Let us see that. So this is the diagrammatic representation of male reproductive system. The diagrammatic view of male reproductive system, a part of the testis open to show inner details, is also given here. So this is the diagrammatic representation. Now let us see the different parts and their functions. The primary sex organs. Remember, the male reproductive system consists the major components such as primary sex organs, primary sex organs, followed by the primary sex organs are followed by accessory ducts, and mainly the various types of glands. So along with this, external genitalia. So these are the very important structural components, of male reproductive system. So now let us focus on each and every component of male reproductive system. First one, the primary sex organs. The primary sex organ mainly includes pair of testes, and the length of the testis, each testis is around 4 to 5 centimeter, and the width is about 2 to 3 centimeter. So these are the testes. So this is one testis, this is another testis. So the testes are located within a sac-like structure called as a scrotum. So scrotum is the sac-like structure within which the testes are located. The very important thing that we should remember is, in both the males as well as in the females, the most of the reproductive organs, the structural components of reproductive system, are located in the cavity called as the pelvic cavity. They are located in the pelvic cavity. So pelvic cavity is nothing but a lower abdominal cavity. So this is also called as lower abdominal cavity. So within this lower abdominal cavity, all the structural components are located. But the very important feature of male reproductive system, that is the location of these testes. Even though these are the primary sex organs, the very important component of the male reproductive system, these are not located within the pelvic cavity. These are not present within the body. These are present as an external organ, but located within the sac-like structure called as scrotum. So that's why the primary sex organs or testes are called as extra abdominal organs. So these are the extra abdominal organs. So the testes are called as extra abdominal organs located within the scrotum. And we will discuss about the scrotum in detail later. But the very important point with respect to the scrotum, that is the importance of this scrotum. You know, the testes are responsible for the production of sperms. Testis is a structure within which the sperms are produced. That's why it is called as primary sex organ. Then what is the importance of scrotum? Scrotum is responsible for the maintenance of temperature, lower temperature. Lower temperature means that is nearly 2 to 2.5 degrees Celsius lesser than body temperature. You know, in a healthy person, the body temperature is around 37 degrees Celsius. But the testis requires nearly 34.5 to 35 degrees Celsius for its function. So that means 34.5 to 35 degrees Celsius means it is less than the body temperature, around 2 to 2.5 degrees Celsius lesser than the body temperature. This lesser temperature is maintained by the scrotum. How it will maintain the lower temperature? So that will be discussed later. Okay. Now let us move to the next important structural components of male reproductive system, that is the accessory ducts. So what about the accessory ducts? The accessory ducts mainly includes the reed testis, vasa efferentia, epididymis, vas deferens, ejaculatory duct, and urethra. See, the very important thing that we should remember is within the testes the sperms are produced, but there is a specific structure for the production of sperms that is called as seminiferous tubules. So seminiferous tubules are the structural and functional units of testes. So these seminiferous tubules open into reed testis. So seminiferous tubules, the name itself indicates these are the tube-like structure. So these tube-like structure within which the sperms are produced, they will open into reed testis. So the seminiferous tubules open into reed testis, and these reed testis, they will open into the next structure called as vasa efferentia. So all the efferentia, vasa efferentia, they are collectively to form a structure called as epididymis. So in this diagram, we can see the epididymis here. So this is the epididymis, and normally the epididymis is located on the posterior surface of each testis. So here is the epididymis and here is the epididymis. And very important thing with respect to the epididymis, that is, in the epididymis, the temporary storage of sperms takes place. The maturation of the sperms takes place in the epididymis and is followed by the epididymis opens into the next tubular structure that is called as vas deferens. And vas deferens is elongated, is emerging from the epididymis, and then it will descend into the abdominal cavity. It will ascend, and then it will enter into the abdominal cavity. So vas deferens is continued as one more structure that is called as the swollen part of the vas deferens, that is called as ampulla. There is a structure called as ampulla, and then ampulla will continue as ejaculatory duct. So in the diagram, here is the ejaculatory duct. So this is the ejaculatory duct. This is the ejaculatory duct. So here you can see the epididymis. Epididymis, they will open into vas deferens. The vas deferens will enter into the abdominal cavity, and then it shows somewhat a swollen part. This swollen part is called as ampulla, and then this ampulla will continue as ejaculatory duct. Remember, this ejaculatory duct is formed by the fusion of this vas deferens, which is continued as ampulla, and the duct coming from a gland called as the seminal vesicle. The duct of the seminal vesicle fused with the ampulla or the vas deferens that will develop as ejaculatory duct. And then ejaculatory duct will continue as an elongated tubular structure which passes through the male copulatory organ that is called as urethra. Remember, urethra is actually emerging from the urinary bladder. It is emerging from the urinary bladder. So this is the duct of urinary bladder. That means it is a part of the excretory system. But the same urethra is connected to the ejaculatory duct also. Ejaculatory duct also. So the urethra coming from the urinary bladder joins with the ejaculatory duct, and that will continue as urethra itself. Remember, urethra is a common passage for urine as well as semen. Urine as well as semen. Now, the next important part of the male reproductive system, that is external genitalia. Then what about this external genitalia? The name itself indicates it is a part of the male reproductive system which is visible externally. Other than the testes, which are located within the scrotum, the another visible structure of male reproductive system that is called as external genitalia. And the external genitalia mainly includes the penis. Penis is nothing but the male copulatory organ, and it consists of an erectile tissue. The penis consists of an erectile tissue. The specific tissue found in the penis which is responsible for the erection of the penis. The name of this erectile tissue is not given in our textbook, but it will be discussed later. So remember, penis is a male copulatory organ, and it consists of an erectile tissue responsible for the erection of the penis at the time of copulation. And the swollen apex, the tip of the penis, that is called as a glans penis, and it is covered by a fold of skin that is called as a foreskin. So this is regarding the external genitalia of male reproductive system. The next very important structural components of male reproductive system called as glands. So these are the glands of a male reproductive system. So the very important question, even in theory as well as in the competitive exams. So what are the glands found in the male reproductive system? What are the secretions of these glands? So the glands again classified into paired glands as well as unpaired glands. And the male reproductive system includes paired seminal vesicles as well as an unpaired prostate gland, and again paired bulbourethral glands. So these are the three types of glands found in the male reproductive system. So the male reproductive system is associated with the three types of glands: paired seminal vesicles, unpaired prostate gland, as well as paired bulbourethral glands. So these bulbourethral glands are also called as Cowper's gland. They are also called as Cowper's gland. And these are mainly responsible for the production of seminal plasma. These glands are mainly responsible for the secretion of seminal plasma. But in that particularly the bulbourethral gland is mainly responsible for the secretion of a fluid which is responsible for the lubrication of penis during the mating process. So during the mating process, before the ejaculation, before the releasing of the semen, there will be the production of a secretion of a fluid which helps in the lubrication of the penis, which avoids the friction between the male copulatory organ and the female copulatory organ. So that is, the process is called as lubrication, and that is because of the bulbourethral gland. And here is the diagram of male reproductive system, and one more diagram is given here. Before that, let me explain important parts. So here is the urinary bladder. Urinary bladder is actually the part of the excretory system, and its duct that is called as urethra. So this is a urethra at the center. But what are the important parts of the male reproductive system? So these are the pair of testes located within the scrotum. So this is sac-like structure is called as scrotum, and each testis consists the structural and functional units called as seminiferous tubules. The seminiferous tubules open into reed testis open into vasa efferentia, vasa efferentia open into epididymis. The epididymis are located on the posterior surface of each testis. The epididymis is the site where the temporary storage of sperms takes place. Epididymis open into vas deferens. Vas deferens enters into the abdominal cavity, and it descends over the ureter or the urinary bladder, and then the vas deferens is continued as a swollen region that is called as ampulla. Ampulla is continued as ejaculatory duct. The ejaculatory duct is formed by the fusion of the duct coming from the seminal vesicle as well as the ampulla, and then it is connected to the urethra, so that it will continue as urethra. And urethra consists an opening that is called as the urethral meatus. The next very important part of the male reproductive system, that is external genitalia. It mainly includes the penis, that is the male copulatory organ. The penis consists of the swollen apex called as glans penis, and is covered by the skin. It is covered by the skin, foreskin, and the glands. There are three types of gland. Here you can see the seminal vesicle. These are the paired glands, 1, 2 paired glands. Then a prostate gland. Here it looks like a paired gland, but this is not the paired gland. So while taking the section, it will appear like this. This is actually the unpaired gland called as prostate gland. The next one is two small bulb-like structures. Here you can see the bulb-like structures. These are called as bulbourethral glands or the corpus glands. So this is regarding the male reproductive system. Now, let us see one more diagram of male reproductive system. Here you can see the front portion. So this is the front portion of the male reproductive system. This is the back portion of the male reproductive system or the male body. So here is the male copulatory organ called as penis, urethra. It is a passage for the common passage for the urine as well as semen. And here you can see the different other structural components. Testis. Here is epididymis. Here is vas deferens. It will continue as ampulla. Then it is joined with the duct of the seminal vesicle. So it will continue as. So this is a ejaculatory duct. Then it joins with the duct coming from the urinary bladder called as urethra. So it will continue as urethra. The very important thing that we should remember is at the junction of this ejaculatory duct and the urethra, there is a presence of prostate gland. And just below the prostate gland, at the neck of this urethra, there are bulbourethral glands. So this is regarding the structure of male reproductive system. Now let us study about the internal structure of testes. The internal structure of testes, the detailed structure. So this is the diagrammatic representation of testes. Actually, if you take the section of the testis, externally it consists in a protective layer. So normally it is made up of different layers. So these are the different layers. So these are the seminiferous tubules. So these are the seminiferous tubules. Here you can see this is the one seminiferous tubule. This is another seminiferous tubules. Say the outermost layer, of the testis, that is called as tunica vaginalis. tunica albuginea, and here is called as tunica vasculosa. So these are the different layers, the dense layers of testis. So each testis is covered by the dense covering, followed by the internal region where the seminiferous tubules, the structural and functional units of testis are located. But before that, see, the entire testis is covered by the dense covering. As I told earlier, it is made up of different layers. And within each testis, there are nearly 250 compartments or the chambers called as the testicular lobules. They are called as testicular lobules. So, the testicular lobule, each testicular lobule consists of 1, 2, 3 highly coiled filamentous structures or tubular structures called as seminiferous tubules. As I mentioned earlier, the seminiferous tubules are the structural and functional units of testis. And each seminiferous tubule consists of two types of cells in their lining. They consist of the lining, each seminiferous tubule lining consists of two types of cells. So the two types of cells are, the first one is called as male germ cells. These are also referred as spermatogonia, as it is given in our textbook. And another type of cells are found in the lining of the seminiferous tubules are called as Sertoli cells. So when compared to the male germ cells, Sertoli cells are larger in size. These are larger in size. So the space between the seminiferous tubule, for example, if this is a one seminiferous tubule, this is another seminiferous tubule, here is one more seminiferous tubule. So this space, the space between the seminiferous tubules are called as interstitial spaces. They are called as interstitial spaces. And these interstitial spaces are characterized by the presence of small blood vessels as well as Leydig cells. Now, see, these Leydig cells, these are also called as interstitial cells. These are also called as interstitial cells. These are the glandular cells. These are responsible for the secretion of male sex hormones. These are responsible for the production of male sex hormones called as androgens. So the androgens are the male sex hormones produced by the Leydig cells. As I told earlier, these male germ cells or the spermatogonia, these are actually the origin of sperms, and these Sertoli cells, they will support the process of sperm production. They will nourish the developing sperms. That's why the Sertoli cells are also called as nurse cells. We will discuss about these Sertoli cells as well as male germ cells in detail while discussing the gametogenesis process. So this is just regarding the structure of, that is the internal structure of testis. Now, so here is the enlarged portion of each seminiferous tubule, the enlarged portion of the seminiferous tubule. Here you can see the lining of the seminiferous tubule. So this is the lining of the seminiferous tubule, and in the first layer you can see the spermatogonium. As it is mentioned in a textbook, these are the male germ cells, spermatogonium. Remember, these spermatogonium are diploid cells, and these spermatogonium later develops as sperms. Here you can see the sperms, the developed sperms. They are also called as spermatozoa, and before that spermatids are formed. So this will be discussed during the gametogenesis, and before that they found in a form called secondary spermatocytes, and the previous step is called as primary spermatocyte. And here is the Sertoli cell, and remember, the Sertoli cells are responsible for nourishing the developing sperm. So this is regarding the diagrammatic sectional view of a seminiferous tubule enlarged. Now, some important points related to testis with the diagrammatic representation. So this is the real photograph of testis. So this is the internal section, the sagittal section of the testis. Now, let us see some additional points. So these are the extra points, that means not given in our NCERT textbook. So these are the points related to the NEET examination. You can expect these questions. They may ask it. The scrotum is a pouch-like structure that hangs behind the penis. So this is a pouch-like structure or the sac-like structure, extra-abdominal structure. So that's why the testes are located outside the body. That's why they're called as extra-abdominal organs. And these scrotum holds and protects the testicles. Remember, testis. So this is singular, and testis, this is plural. So this is a difference between the term testis as well as testis. Now, the next important point is it contains numerous nerves and blood vessels. The scrotum is provided with numerous nerves and blood vessels. And during the times of lower temperature, I told at the beginning itself, the scrotum is responsible for the maintenance of lower temperature. Now the question arises, how this scrotum maintains a lower temperature? You know the homeostasis mechanism found in our body. During the cold condition, the shivering takes place that maintains the constant body temperature. And when there is a hot condition, the sweating process takes place that again regulates the body temperature. So that the constant body temperature is maintained, that is a 37 degrees Celsius. But the temperature required for the proper functioning of the testis is lesser than that. That is nearly 34.5 to 35 degrees Celsius. That is 2 to 2.5 degrees Celsius lesser than that of body temperature. And this lower temperature is maintained by the help of scrotum. So not only the lower temperature, even that helps in the maintenance, so the regulation of that constant temperature. How it is regulated? See here, during the times of lower temperature, when there is a cold climate, when there is a cold condition, there is a muscle called as cremaster muscle. This cremaster muscle contracts and pulls the scrotum closer to the body, while the another muscle called as a darter's muscle gives it a wrinkled appearance. So when there is a cold condition, what happens? The cremaster muscle found in the scrotum undergoes contraction so that it pulls the scrotum closer to the body. When the scrotum comes closer to the body, the testis will come closer to the body so that it will absorb the heat energy from the body, so that the constant temperature is maintained. Because during the cold climate, the temperature falls lesser than 34.5 degrees Celsius in the testis. So there is a need to maintain 34.5 to 35 degrees Celsius temperature. So some amount of temperature, heat energy is required. So that heat energy is absorbed by the body by the help of scrotum. So during this condition, during the lower temperature, what happens? The cremaster muscle contracts and pulls the scrotum closer to the body, and it helps in absorption of heat energy from the body. At the same time, while the darter's muscle, the another type of muscle, darter's muscle, gives it a wrinkled appearance, Then, when the temperature increases, when there is a hot condition, when the temperature increases, the cremaster muscle and the darter's muscles relax to bring down the scrotum away from the body and remove the wrinkles respectively. Cremaster muscle undergoes relaxation so that the scrotum move away from the body, so that testis will move away from the body surface. The temperature reduces, means it will be maintained at 34.5 to 35 degrees Celsius. At the same time, the relaxation of these darter's muscle removes the wrinkles formed in the testis. So this is regarding the functioning of the scrotum. So the scrotum remains connected with the abdominal cavity or the pelvic cavity by a canal called as inguinal canal. So this is another very important point that should be remembered. The scrotum remains connected with the abdominal cavity or the pelvic cavity by inguinal canal. Now, some additional points related to the mechanism of descending of testis into the scrotum. So normally the testis descend in the scrotum sac when the fetus is about 7 months old. So during the embryonic development, it will be there within the pelvic region. But when the fetus is around the 7 months old, the testis descend into the scrotal sac. And it occurs under the influence of hormones called as FSH as well as the testosterone. FSH and testosterone. If they fail to descend, sometime there is a failure of descending of testis into the scrotal sac. If they fail to descend, it is called as cryptorchidism. That is called as cryptorchidism, and it leads to sterility. Because when the testis is not descend into the scrotum, it will remain within the body. And you know the body temperature is 37 degrees Celsius, which is unfavorable for the functioning of testes. When they descend into the scrotum helps to maintain the lower temperature so that the testis becomes functional. So when there is a cryptorchidism, such male individual becomes sterility because of this reason. So testes are surrounded by 3 layers of tissues. As I mentioned earlier, the 3 layers are, the first one is tunica vaginalis. This is the outer covering and is followed by tunica albuginea. So this is a middle fibrous layer. This is a middle fibrous layer. And the next one is tunica vascularis. So this is the innermost region, and this is the inner network of capillaries. It is highly vascularized. That's why it is named as tunica vascularis. The next, some additional points related to the penis. There is a male copulatory organ. Say the penis consists 3 cylindrical cords of spongy erectile tissue. So in our textbook, the presence of erectile tissue is mentioned, but the name of that erectile tissue is not given. Actually, the penis consists of 3 spongy erectile tissues or the cavernous tissues. That is 2 cords of corpora cavernosa. Total 3 cords, 2 cords of corpora cavernosa and 1 cord of corpus urethrae. See, the corpus urethrae is also called as corpus spongiosum. So most commonly this corpus spongiosum is mentioned. Additional to the corpus spongiosum, that is also called as corpus urethrae, 2 more cords of erectile tissues are there. They are corpora cavernosa. So this is the very important point related to the penis, that is regarding the erectile tissue. And see the foreskin, this is the skin which is covering the penis, that also covers the tip region of the penis. And that skin is called as foreskin. And normally this foreskin, it will move backward during the erection of the penis. In some individuals, it may not move backward during the coitus or during the mating process. In such case, the foreskin is removed surgically by the process called as circumcision. That is called as circumcision. So circumcision is nothing but the removal of the foreskin. So in the individuals where there is no backward movement of foreskin during the erection of the penis. Okay, so the foreskin is also called as a prepuce. So this is another point to be remembered. The foreskin is also called as prepuce. And see in the penis region there is a glandular structure. So they are called as prepucial glands. Prepucial glands. These prepucial glands present in the skin of the penis neck. So penis neck means if it is a glans penis, if it is a gland penis, the basal part. So this is a tip region. So the basal part in this region, so there is a presence of a prepucial gland, and this prepucial gland is responsible for the secretion of white sebaceous substance, called as smegma. So that is called as smegma. So that is a very important term. You may expect this. So this is an additional point. Smegma is nothing but a white sebaceous substance secreted by prepucial glands present in the skin of the penis neck. And the microbial infection in smegma cause irritation due to inflammation. There is no specific positive significance to this, but the microbial infection in this smegma, it causes the irritation due to the inflammation. So this is an additional point. So now let us move to study about the female reproductive system, the structure of female reproductive system. So this is the diagrammatic representation of female reproductive system. So before that, let me mention the very important structural components of female reproductive system. As I mentioned in the male reproductive system, the female reproductive system also includes primary sex organs. So primary sex organs mainly includes a pair of ovaries, and it is included with the female reproductive system also consists of accessory ducts, and the accessory ducts includes fallopian tube and urethra, followed by cervix as well as vagina. So these are the different structural components. And the next very important component, external genitalia. External genitalia. And finally, the associated one, the associated glands, the glands of female reproductive system. So these are the major structural components of female reproductive system. As I told earlier, all the structural components of reproductive system are located in the pelvic region. In male individuals, the testes are located externally in the scrotum, that is the extra abdominal organ. But in female reproductive system, almost all the structural components are located within the pelvic region or lower abdominal cavity. It is also called as lower abdominal cavity. So pelvic region or the lower abdominal cavity consists of all the structural components of female reproductive system. It mainly includes the primary sex organs. As I mentioned earlier, it includes pair of ovaries. So these are ovaries are located on either side of the pelvic region, and each ovary has a 2 to 4 centimeter length. The length of each ovary is around 2 to 4 centimeter. And these ovaries are located in their position by the help of ligaments. So they are attached to the uterine wall at one side and pelvic wall at another side by the help of ligaments. And internally, the inner region of the ovary is called as ovarian stroma. The inner region of the ovary is called as ovarian stroma. It will be discussed later in detail. And the ovarian stroma is again differentiated into outer cortex and inner medulla, within which the eggs are produced. So ovary is a primary sex organ responsible for the production of egg or the ovum. So as well as it is also involved in the production of some ovarian hormones. So even the testis is also involved in the sex hormones. That's why the testis and ovary are also considered as endocrine glands, since they produce hormones. Here ovarian hormones are produced by the ovary. The next structural components of female reproductive system, there is accessory ducts, and these accessory ducts are also called as associated ducts, responsible for the transportation of gametes mainly. It includes the first one, oviducts. See, oviducts are also called as fallopian tubes. So these are also called as the fallopian tube. Oviducts are also called as fallopian tubes. And it has around 10 to 12 centimeter length. The length of the oviduct is around 10 to 12 centimeter. And it also has different parts. See here, this is one oviduct, this is another oviduct. So the funnel shaped region of the oviduct is called as infundibulum. And this infundibulum consists of small finger-like projections. See, these finger-like projections are called as fimbriae. They are called as fimbriae. Remember, these fimbriae are responsible for the collection of ovum during the process of ovulation. And the swollen region of the oviduct or fallopian tube are called as ampullum. The same term even used in case of male reproductive system. The swollen part of the vas deferens is called as ampulla. Here, swollen part of the oviduct are called as ampulla. And the last part of the fallopian tube, which is connected to, which is attached to the, or which is present near to the uterus. So this is the uterus. The part of the oviduct which is attached to the uterus is called as isthmus. That is called as isthmus. And the junction between the ampulla and isthmus, that is called as ampullary isthmic junction. That is called as ampullary isthmic junction. So this ampullary isthmic junction is a very important part. This is also a very important part, upper fallopian tube, where the fertilization process takes place. Now, the next important point, structural component of accessory duct, that is uterus. So uterus is also known as womb. It is generally known as womb, the very important part. Each and every part is important, but it is a very important part at the time of reproduction, because the development of the child takes place inside the uterus. And uterus has an inverted pear shape. This is an inverted pear shape, and it is attached or connected with the ligaments. It provides support. And the very important point with respect to the uterus, that is the presence of uterine wall. The structural components of uterine wall. See, uterus has uterine wall, and the uterine wall is made up of 3 layers. Uterine wall is made up of 3 layers. One is outermost perimetrium, followed by middle myometrium, and innermost endometrium. So perimetrium is the outer lining. It is the outer lining, and myometrium is made up of smooth muscles. It is mainly made up of smooth muscles. And endometrium is made up of glandular cells. See, endometrium is made up of glandular cells. The very important point regarding endometrium, that is, the tissue of endometrium has a capacity to undergo degeneration as well as regeneration. It has the capacity to undergo degeneration as well as regeneration. So this is regarding the different layers of uterine wall. And one more thing that I should mention here, perimetrium is just providing the protection as an outer lining. It provides protection. And myometrium, it is made up of smooth muscles. It is mainly responsible for the contraction. It is mainly responsible for the contraction at the time of childbirth. At the time of childbirth. So during the parturition, the contraction of uterine wall is mainly because of the myometrium. And endometrium is the site of implantation. It is a site of implantation. So implantation is nothing but the attachment of blastocyst. So the site of implantation, that is the endometrium, and the degeneration of this endometrium results in the menstruation. So that will be discussed later again. So now the next important structural component of female reproductive system as given in our textbook, that is external genitalia. Say external genitalia, this is the external visible part of the female reproductive system. Before that, let me add one more point, that is to the accessory duct, that is vagina. So one more very important component of the accessory duct, vagina. See here, vagina is a narrow passage. It is a narrow passage into which the penis is inserted at the time of copulation. So in this region, the releasing of semen takes place, then the sperms will enter into the next region. So that is called a cervix. So cervix is the narrow region. It is the neck region of the uterus. It is the neck region of the uterus. And cervix has a passage, a narrow passage, that is called a cervical canal. That is called a cervical canal. And the opening of this cervix is called as a cervical orifice. That is called a cervical orifice. And this cervical canal along with the vagina and cervical canal together known as birth canal, through which the child will come out at the time of parturition. So now let us move to the next one, that is external genitalia. See, the external genitalia, the visible components or the visible parts of the female reproductive system, it mainly includes the mons pubis and the labia majora, labia minora, hymen and clitoris. So what is this mons pubis? It is a fatty cushion-like structure covered by the skin and pubic hairs. It is the front portion of the female body. It is a cushion-like fatty tissue covered by the skin and the pubic hairs. And labia majora. Labia majora is the fold of skin. It is a skin folding externally present. So it covers the opening of the female reproductive system, that is called as labia majora. And the inner skin foldings called as labia minora. So outer skin foldings are pair of skin folding called as labia majora, and inner skin foldings are called as labia minora. See, at the junction of labia minora, at the junction of upper junction of labia minora, there is a small finger-like projection that is called as a clitoris. The small finger-like projection that is called as clitoris. Clitoris is just like the erectile tissue of a male reproductive system of the penis. That means clitoris is highly touch sensitive. So it is responsible for the sexual excitement in the female individuals. Then about the hymen, hymen is a thin layer that partially covers the vaginal orifice, opening of the vagina. So normally that will be torn during the first coitus, or even in the individual, those who are involved in the physical exercises like cycling or the dancing, or sometimes even because of the insertion of the different types of tools, sex toys. So that also leads to the breakdown of this hymen. So this is regarding the the female reproductive system, the important points given in our textbook. So one more diagrammatic representation is there. Before that, let me explain this. See, this is a ovary. These are the pair of ovaries. The ovaries are connected to the pelvic wall and the uterine wall by the help of ligaments. So these are the ligaments. And here is the oviduct or the fallopian tube. And I told about the different parts. This is the funnel part, funnel shaped part called as infundibulum, finger-like projections called as the fimbriae, the swollen part called as ampulla. The next ampular ischemic junction and isthmus. And this inverted pear-shaped structure is called as uterus. And this dome-shaped part of the uterus is called as uterine fundus. The cavity present within the uterus is called as uterine cavity. And then wall of the uterus, there are three wall layers: outer perimetrium, middle myometrium, innermost endometrium. And then narrow neck region of the uterus, that is called as the cervix. The neck region of the uterus is called as cervix. Within the cervix, there is a narrow passage. This narrow passage is called a cervical canal. The opening of the cervical canal is called a cervical orifice. So the cervical orifice open into a narrow passage that is called as vagina. So till here, the penis will come at the time of mating process. The releasing of semen takes place in this vagina. So this vagina and the cervical canal together known as birth canal, through which the child will come out at the time of parturition. So this is the diagrammatic representation given in our textbook, the very important diagram for the theory exam. And here is the one more diagrammatic representation. So this is the side view of a female reproductive system. This is the front portion. This is the back portion of a female body. And here you can see the very important parts such as the pubic symphysis. See, this portion is called as mons pubis. So mons pubis is the front portion. So this cushion-like fatty tissue. So it is covered by the skin, and here the hairs are found. So this is regarding the pubic symphysis, the mons pubis, the front portion of the female body. And then there is a passage, there is a common opening. There is a particular term to this aperture. So it will be discussed later. And within which there are two openings. One is the opening of urethra. So just remember, in case of females, there are separate openings for the releasing of urine as well as for the insertion of penis. The passage for the insertion of penis, that is called as vagina. So this is the vaginal opening. And here is the opening of the urethra, urethral meatus, urethral orifice. So this is a vaginal orifice. So both the openings will open into the common aperture. Common aperture. So this is a vagina, and here is the uterus. So this is the uterus. So that means this narrow region is called as a cervix. It will open into the uterus. It will open into the uterus. And here is the opening. There is the opening of the anus. So that is related to the digestive tract. So this is regarding the reproductive system of a female reproductive system. And one more point, here is the structure, the small finger-like projection called as clitoris, the highly touch-sensitive structure of female reproductive system. Now, this is the real photograph of female reproductive system. That means the internal structures. Here you can see the ovaries. So this is the ovaries, fallopian tube. And here you can see the releasing of ovum. See this structure is ovum. And this is the ovary, the releasing of ovum. So the process of removal, releasing of ovum from the ovary is called as ovulation. Right. So this is a single ovum and ovary. So this is the internal structure of the ovary. As I mentioned earlier, the ovary is also covered by the protective layers. You can see the different structures labeled here. The very important parts will be discussed now. Say this is a lining, the outer covering of the ovary, somewhat spherical shaped structure, and these are located in their position by the help of ligaments. And each ovary is provided with the blood vessels, and the inner region of the ovary is called as stroma. The inner region of the ovary is called as stroma, and the stroma is differentiated into outer cortex and inner medulla. So in the cortex region, in the outer cortex region, the development of ovarian follicle takes place. So these are the ovarian follicle. In that, this is a primary follicle followed by secondary follicle, tertiary follicle. Here you can see the tertiary follicle. This is a matured follicle. This is called as Graafian follicle. So this is a Graafian follicle, and after the ovulation, it becomes an empty Graafian follicle that is specifically known as corpus luteum. And if there is no fertilization, the corpus luteum also degenerates and it forms the corpus albicans, and finally it undergoes degeneration. So that means the different steps of ovarian follicle development, or the Graafian follicle development, and the later stages are mentioned in this diagrammatic representation. So just remember, the eggs are produced within the structural and functional units called as the Graafian follicle. So this is a structure of a single Graafian follicle. The egg producing structure within the ovary, just like the seminiferous tubules of testis. In the ovary, the structural and functional units are the Graafian follicle or ovarian follicles. See, the Graafian follicle is externally covered by the protective layers called as the theca externa followed by theca interna, and is followed by the membrana granulosa. So theca externa followed by theca interna followed by membrana granulosa. Here you can see the granulosa cells, and here is a stalk-like structure. Internally there is a stalk-like structure. That the stalk-like structure is called as the cumulus oophorus. So this cumulus oophorus holds the female gamete, that is the egg cell. But in case of human beings, specifically in this Graafian follicle, the egg is found in a secondary oocyte stage. That means that is yet to complete the second meiosis. So this is the cumulus oophorus, and then there is a layer called as a corona radiata. The corona radiata is followed by non-cellular protective layer called as zona pellucida, and then internally it consists of secondary oocyte. So secondary oocyte is nothing but the cell that consists its own membrane, that is a plasma membrane. In egg it is specifically known as ulemma. So here you can see the different structural components: theca externa, theca interna, membrana granulosa, followed by cumulus oophorus. So here is theca externa, theca interna, cumulus membrana granulosa, followed by cumulus oophorus, and then corona radiata, and followed by the zona pellucida. So this is the corona radiata. Zona pellucida is present inner to the corona radiata, and followed by the secondary oocyte. So internally there is a secondary oocyte. That means it is yet to complete the second meiosis. And another very important thing, here you can see the space. So this is the space present within the Graafian follicle. This is called as antrum. The space is called as antrum. And this space is filled with liquid folliculi. The liquid folliculi is present inside this antrum. So this is regarding the structure of Graafian follicle or ovarian follicle. Then why it is named as Graafian follicle? Why it is called as Graafian follicle? Because of the scientist named as de Graaf, who described this structure in 1672. So this is regarding the Graafian follicle. Now let us see some additional points about vulva. Then what is meant by vulva? Vulva is nothing but external genitalia. This is nothing but external genitalia. So external genitalia of a female individual is called as vulva. So it has a depression, the common opening to which the urethral orifice as well as vaginal orifice are open. That common depression, common opening of female reproductive system, that is called as vestibule. That is called as vestibule, and that is present in front of the anus. So the vestibule has two apertures, as I mentioned earlier. The upper external urethral orifice of urethra, and the lower opening, that is called as vaginal orifice of vagina. So through this urethral orifice, the urine will come out, and through this vaginal orifice, the penis will be inserted into the vagina. The vestibule is bounded by two pairs of moist skin folds with the sebaceous glands. The inner small pair called as labia minora, and outer larger pair is called as labia majora. Remember, labia majora is homologous to scrotum. So this is another very important point to be remembered. Labia majora is homologous to scrotum, just like the scrotum of a male reproductive system. So labia minora fuse anteriorly. Labia minora fuse anteriorly to form a skin fold called as prepuce. So the term prepuce is even used in case of female reproductive system. Remember, labia minora fuse anteriorly to form the skin fold called as prepuce in front of the small erectile organ called as clitoris. And this clitoris is homologous to penis. So that is another important point to be remembered. Clitoris is homologous to the penis. Why? Because both are supported by corpora cavernosa. The erectile tissue, corpora cavernosa, are formed in. The next important point, the labia minora also fuse posteriorly. The labia minora also fuse posteriorly to form the membranous fold called as a fourchette. So that is called as a fourchette. So fourchette is the labia minora fused posteriorly to form a membranous fold. And the area between the fourchette and the anus is called as perineum. So perineum is nothing but the area between the fourchette and the anus. And there is a fleshy elevation. There is a fleshy elevation above the labia majora and known as mons veneris. Remember, mons veneris is actually known as mons pubis. This term is given in the textbook. But remember, mons pubis is also known as mons veneris, which has pubic hairs. The development of pubic hair is one of the secondary sexual character of female individual. So these are the very important points related to vulva, that is a female external genitalia. So this is a diagrammatic representation, shows all the points what we discussed in the previous slide. So this is a common aperture. So this is a common aperture of female reproductive system. This common aperture of depression is called as a vestibule. So within the vestibule, two more openings are there. So one is the opening of vagina, that is called as vaginal orifice or vaginal entrance. The another small opening that is called as urethral orifice or the urethral opening. So the outer skin folding, here you can see the outer skin folding. This outer skin folding, this is the inner skin folding, here is the outer skin folding. So the outer skin folding is called as labia majora. So this outer skin folding is called as labia majora, and this inner skin folding is called as labia minora. So at the junction of labia minora, this is the junction of labia minora. At the junction of labia minora, there is a small finger-like structure that is called as clitoris. The glands of clitoris is located here. Okay, and the one more important point, that is the prepuce of clitoris. It is a fold of skin. It is formed due to the joining of the fusion of labia minora. And this is the front portion of a female body, that is called as mons pubis. So this is a mons pubis. Here you can see the pubic hairs. These are the pubic hairs. And the region between this posterior fourchette and the anus, this particular region is called as perineum. So this is regarding the different parts of vulva or the external genitalia. Now, another very important component of female reproductive system, the associated structure called as mammary gland. But in human beings, the mammary gland is not located in the pelvic region. Then what are these mammary glands? So this is the diagrammatic representation of mammary gland. Say mammary glands are commonly known as breast. So mammary gland is commonly known as breast or mammary glands, pair of breast or the pair of mammary glands. So these are basically the modified sweat glands. Okay. Now let us see the important points. See, the presence of mammary gland is the characteristic features of mammals. Mammalia. Mammary glands are found both in males as well as in the females, but functional in females. Mammary glands are functional in females. The presence of functional mammary glands is a unique feature of female mammals, and these are the paired structures commonly known as breast. And these breast or the mammary glands consist glandular tissue as well as variable amount of fat. See, these glandular tissue are mainly responsible for the secretion of milk. Then what about this variable amount of fat? See, based on the amount of fat, the size and shape of the breast varies. Based on the amount of fat found in the breast, the size as well as the shape varies from individual to individual. Internally, each mammary gland consists of 50 to 20 mammary lobes. So 15 to 20 chambers present within each breast or the mammary gland are called as mammary lobes. And these mammary lobes consist a group of or a bunch of small sac-like structures called as alveoli, or these are specifically known as mammary alveoli. Remember, these mammary alveoli are the structural and functional units of mammary glands because they secrete, these mammary glands, they secretes the milk. The secretion, production of milk takes place in the alveoli. So they secrete the milk as well as they store the milk in the lumen. And these milk secreted within the alveoli are then transported through the ducts. They are transported through the ducts. The various types of ducts are there. Just imagine if it is the alveoli, each alveoli consists a duct. So this is the duct of the alveoli. This duct of alveoli, each alveoli is called as mammary tubule. That is called as mammary tubule. So now, these mammary tubules, then they will open into mammary duct. They will open into mammary duct. And mammary duct are then developed as, or they will continue as mammary ampulla. Mammary ampulla are followed by lactiferous duct. That means mammary ampulla again narrows and they will continue as lactiferous duct. And all the lactiferous ducts are joined together at one point. That leads the formation of a structure called as nipple. That is called as nipple. So this is regarding the internal structure. I will show the diagrammatic representation of different ducts here. So these are the alveoli, and each alveoli consists of duct called as mammary tubule. And these mammary tubules, they are joined together. They are joined together to form, the mammary tubules are joined together to form mammary duct. And these mammary duct then undergoes enlargement. The enlarged portion of a mammary duct is called as ampulla. And then these ampulla are again continued as narrow tubular structure called as lactiferous duct. So alveoli followed by mammary tubule, followed by mammary duct, followed by ampulla, followed by lactiferous duct. So many number of lactiferous duct, they are grouped at the tip of the mammary gland. So that leads to the formation of a nipple. So nipple is the multiporous structure. Remember, nipple is the multiporous structure. This is not the uniporous structure, multiporous structure, because each lactiferous tubule consists an opening. So that's why it is a multiporous structure. And this nipple, the region of nipple, it is covered by a thick dark colored skin. So around the nipple there is a thick region. It is covered by thick dark colored skin. So this thick region surrounding the nipple region is called as areola. So this region is called as areola. This areola is characterized by the presence of oil glands. It will secrete the oil, which helps in avoiding the friction at the time of breastfeeding. It avoids the friction during the breastfeeding, during the suction of milk by the child. The friction will, the pain may cause. The pain is avoided because of the secretion of the glands, the oil glands found in this area. So this is regarding the structure of mammary gland. Now, let us move to the next very important mechanism in human reproduction, that is gametogenesis. So what is meant by gametogenesis? In simple words, gametogenesis is nothing but the production of gametes. But before production of gametes, some events takes place. That is the formation. So the formation of gametes, and after the formation of gametes, they should become functional. So that process is called as maturation. So the formation and maturation of gametes together known as gametogenesis. So what is mean by gametogenesis? The process of production of male gametes, that is sperms, and female gametes called as eggs in the primary sex organs called the testis and ovaries respectively is known as gametogenesis. Sperms within the testis and eggs within the ovary called as gametogenesis. As I mentioned earlier, there are two types. One is the spermatogenesis. Spermatogenesis is nothing but the process of production and formation of male gametes or the sperms in the testis. And the another one is oogenesis, nothing but the process of production and formation of female gametes or ovum or eggs in the ovary. So these are the types of gametogenesis. Now let us discuss about the types of gametogenesis, that is spermatogenesis and oogenesis in detail. So here is the diagrammatic representation of spermatogenesis. Just to see the diagrammatic representation, the starting point of a spermatogenesis is male germ cells. But before that, let me tell you the very important feature of male individuals. Normally, in both males as well as in the females, the gametogenesis begins only after attaining the sexual maturity, that is called as puberty. So only after attaining the puberty, both the male individual as well as female individual start to produce the gametes, start to release or produce the gametes. So this is called as sexual maturity or the puberty. So in case of a male individual, normally the male individual reaches the puberty at the age of 50. So at the age of 15th year, the male individual attains a sexual maturity. Sometimes it may vary, 14 to 15. At this age, the male individual start to produce the gametes. So when the male individual attains the sexual maturity, the gametogenesis process begins. Remember, the beginning of gametogenesis takes place, that is spermatogenesis takes place only after attaining puberty in male individuals. So this is a very important difference between the male and female individual. I told the beginning takes place only after attaining puberty in case of male individuals. So the starting point of gametogenesis, that is the spermatogenesis, that is the male germ cells. As I mentioned earlier, testis is the structure within which the sperms are produced. But in the testis, particularly the seminiferous tubules are the structural and functional units. Seminiferous tubules are the structural and functional units within which the sperms are produced. But you know, the seminiferous tubules consist of two types of cells. Male germ cells as well as sertoli cells. So here, male germ cells develop as the origin of sperms. Male germ cells acts as an origin of sperms. And sertoli cells, they will support the process of spermatogenesis. Support means they will provide nourishment. They nourishes the developing sperms. So now, in our textbook, the male germ cells also mentioned as spermatogonia. They are also mentioned as spermatogonia, as given in our textbook. Okay, let us consider it as spermatogonia. So now what happens? These spermatogonia, basically the diploid cells, these spermatogonia undergoes a multiplication phase. See, the phase of multiplication is nothing but mitotic division. The continuous mitosis in the spermatogonia or the male germ cells leads to the production of a large number of spermatogonia. It leads to the production of large number of spermatogonia. It produces large number of spermatogonia. And these spermatogonia are classified into two types. Type A spermatogonia as well as type B spermatogonia. So what is the difference between, what are the differences between this type A spermatogonia and type B spermatogonia? Remember, type A spermatogonia will continue the process of mitotic cell division. And this mitotic cell division again leads to the production of some more number of spermatogonia. Remember, type A spermatogonia, type B spermatogonia are produced by the mitotic division of spermatogonia itself. But type A spermatogonia will continue the process of mitotic division, but type B spermatogonia will continue. Type B spermatogonia will continue the process of, they will continue the process of spermatogenesis. They will continue the process of spermatogenesis. So remember, only the type B spermatogonia develops as sperms. They will develop as sperms. How? See, this type B spermatogonia is also diploid because it is produced by the mitotic division. Now, this type B spermatogonia undergoes cell enlargement. It undergoes cell enlargement. Cell enlargement means increase in the size. So nothing but increase in the size. Increase in the size takes place due to the accumulation of reserve growth. See, this is called as growth phase. Increase in the size is called as growth. So this is called as growth phase, so that there is a difference in the size of the cell. Here smaller size, here it is larger in size. And this cell produced after the growth phase is known as primary spermatocyte. It is called as primary spermatocyte, and this is also diploid. There is no change in the chromosome number, just to change in the size. No division, just change in the size. Now, this primary spermatocyte undergoes meiosis 1. What is this meiosis 1? Actually, after the completion of growth phase, the next phase is called as maturation phase. So what is this maturation phase? Maturation phase is nothing but meiosis, during which the meiosis takes place. But you know, the meiosis takes place in two steps: meiosis 1, meiosis 2. The meiosis 1 leads to the production of two daughter cells. Meiosis 1 in the primary spermatocytes leads to the formation of two daughter cells. And these two daughter cells are called as secondary spermatocytes. And both these secondary spermatocytes are haploid. These are haploid. Why these are haploid? Because meiosis 1 is nothing but reductional division. This is nothing but reductional division. If the parental cell consists of 46 chromosomes, after the meiosis 1, after the meiotic 1, it consists 23 chromosomes. This also consists 23 chromosomes. Okay, so now the daughter cells are haploid cells, and these are called as secondary spermatocytes. Now, both the secondary spermatocytes undergoes meiosis 2. Now it undergoes meiosis 2. That means second step in meiosis. Only one meiosis. In that second step, meiosis 2. So meiosis 2 leads to the formation of total 4 haploid cells. And all the haploid cells are spherical in shape. All the haploid cells are spherical in shape. Non-motile, non-functional, inactive, spherical shaped cells are formed. And these spherical shaped cells formed at the end of meiosis 2 or at the end of the maturation phase are called as spermatids. Remember, the spermatids. Spermatids are spherical. The spermatids are spherical. Then non-motile, they cannot move, they are non-motile, and inactive, they are inactive. And these are haploid cells. These are haploid cells. So both the primary spermatocytes, secondary spermatocytes. So this is a primary spermatocyte. After the meiosis 1, these are the secondary spermatocytes. Produces totally 4 haploid cells. So these are called as spermatids. And all these spermatids are haploid. These are haploid. Okay. So this is actually the first step in spermatogenesis. Till here, till the production of spermatids, one step, formation of spermatid. So after the formation of spermatid, the next very important event that is called as spermiogenesis. In spermatogenesis, first step is the formation of spermatid, followed by the second step that is called spermiogenesis. See the difference between spermatogenesis and spermiogenesis. Spermiogenesis is a step in spermatogenesis. Then what is meant by spermiogenesis? See, spermiogenesis is nothing but the production of sperms. See, the sperms are also called as spermatozoa. The production of sperms from spermatids. Production of sperm means actually the transformation takes place. The conversion takes place. The transformation of spermatids into sperms is called as spermiogenesis. That is called as spermiogenesis. The process of conversion of spermatid into spermatids is called as spermiogenesis. So it is also defined as the transformation or the conversion of spherical, non-motile, inactive, non-functional spermatids into tadpole-shaped, motile, active, functional sperms is called as spermiogenesis. That is called as spermiogenesis. So this is regarding the spermatogenesis. So one spermatogonia totally produces 4 sperms at the end of the spermatogenesis. From one spermatogonia, one type B spermatogonia, specifically type B spermatogonia, or from one primary spermatocyte, 4 sperms are produced, and all these sperms are haploid. All these sperms are haploid. Let us see the important points. So the production and formation of male gametes or sperms in the testis is called as spermatogenesis, during which the immature male germ cells or spermatogonia are the starting point. They produces sperms, and these immature male germ cells, spermatogonia, produces sperms at the end of the spermatogenesis. But this process begins only at the puberty in male individuals. So in males, the spermatogenesis begins at maturity, at puberty. So during which the spermatogonia multiply by mitotic division and increase in the numbers, produces type A and type B spermatogonia. And the type B spermatogonia undergoes cell enlargement that leads to the formation of primary spermatocytes. And these primary spermatocytes undergoes meiosis. And the meiosis leads to the production of secondary spermatocytes, that is first meiosis. And secondary spermatocytes undergoes second meiosis, leads to the formation of spermatids. So the first meiotic division, that is a reductional division, produces 2 haploid secondary spermatocytes, and then second meiotic division produces 4 haploid spermatids. And then spermiogenesis takes place, that is nothing but the conversion of spermatids into spermatozoa or sperms. And finally, the matured sperms are released into the lumen of a seminiferous tubule from Sertoli cells. From Sertoli cells is called as spermiation. Then what is the importance of Sertoli cells? Remember, after the production of spermatids, these spermatids are embedded. These spermatids are embedded in Sertoli cells to produce sperms. So remember, the spermatids are embedded in the Sertoli cells, where they will develop as sperms, during which the Sertoli cells provides nourishment to the developing sperms. Now let us see the hormonal regulation of spermatogenesis. So all the reproductive processes are controlled by the hormones. So there is a significant increase in the secretion of GnRH, nothing but gonadotropin releasing hormone, and it is a hypothalamic hormone at puberty. So when the male individual attains the sexual maturity or the puberty, there is a significant increase in the secretion of gonadotropin releasing hormone called as GnRH. Remember, it is a hypothalamic hormone, means it is secreted by the hypothalamus. The increased levels of gonadotropin releasing hormone induces anterior pituitary gland to produce two types of gonadotropins, that is luteinizing hormone or LH and follicle stimulating hormone called as FSH. So first, gonadotropin releasing hormone secreted by hypothalamus that induces anterior pituitary to produce two gonadotropins, that is luteinizing hormone and follicle stimulating hormone. Now, the luteinizing hormone induces the Leydig cells or the interstitial cells present in the interstitial space within the testis, that is between the seminiferous tubules, called Leydig cells. So the LH induces Leydig cells to produce the male sex hormones called as androgens. Then androgens are responsible for regulating the mechanism of spermatogenesis. At the same time, the follicular stimulating hormone induces Sertoli cells, the nourishing cells, to produce some factors responsible for or required for spermiogenesis. That is the last step in spermatogenesis, that is the transformation of spermatids into sperms. For that, some factors are required. Such factors are secreted by the Sertoli cells. You know, the Sertoli cells. In the Sertoli cells, the spermiogenesis takes place. And after the completion of this process, the sperms are released into the lumen of the seminiferous tubule by the process called as spermiation. Now, let us study the structure of human sperm. The structure of human sperm. Here is the diagrammatic representation of human sperm. This is a matured human sperm, tadpole-shaped structure. It consists of different parts. Remember, this is a cell. This is a cell. So it must contain the cell membrane. So there is a cell membrane or the plasma membrane. The outer body of the sperm is covered by the cell membrane or the plasma membrane. Just see the different parts. Say the human sperm is differentiated into three major parts. One is called head. Another one is middle piece. Before that, there is a neck, a middle piece, and a tail. So these are the major parts of the human sperm. So head is the anterior segment. Head is the anterior segment. So this is the head portion, anterior segment, and it is oval in shape, and it consists of paternal haploid nucleus. So this is a very important part of the sperm. This is the haploid nucleus. Haploid means it consists 23 chromosomes. 23 chromosomes, not 23 base, 23 chromosomes. And this is called as a paternal chromosome. It is called as paternal chromosome. So it consists of nucleus. The nucleus consists of a haploid set of chromosomes. And the sperm head, it is externally or at the apex region, it is covered by a cap-like structure. It is covered by a cap-like structure that is called as acrosome. And this acrosome consists of hydrolytic enzymes. Three types of hydrolytic enzymes: acrosin, hyaluronidase, as well as neuraminidase. Hyaluronidase, acrosin, and neuraminidase. So these are the three important type of enzymes found in the acrosome, or the proteinase enzymes. See, these enzymes, hydrolytic enzymes, are responsible for the penetration of sperm into the ovum. The penetration of sperm into ovum takes place by the help of these hydrolyzing or hydrolytic enzymes. They will break the protective layers of egg so that it helps for the entry of sperm into the ovum. And one more thing, this acrosome is derived from the Golgi apparatus. It is derived from the Golgi apparatus or the Golgi zone. Now, the next part of the human sperm, that is neck. Neck is the narrow region which connects the head to the middle piece. Head to the middle piece. So this is the neck, narrow region of the human sperm. And the neck region consists of proximal centriole. There are two types of centrioles, proximal centriole as well as distal centriole. Remember, the neck region consists of proximal centriole. And these proximal centrioles are responsible for the production of spindle fibers at the time of cell division. The next major part of the human sperm, there is a middle piece. And the middle piece consists of distal centriole. And this is distal centriole produces the axial filament. and there is a double row of mitochondrial sheath. There is a double row of mitochondrial sheath. You can see these are the mitochondrial sheath. So the mitochondrial sheath, mitochondria is nothing but the energy producing structure cell organelle, the powerhouse of the cell. So it is also called as nebenken. The nebenken is nothing but the mitochondria of the human sperm present in the middle piece. So the double row of mitochondrial sheath called as nebenken is found in the middle piece. So produces energy required for the movement of the sperm. So that's why it acts as the engine room of the human sperm. And axial filament, this is the tail part, axial filament. This axial filament is originated from the distal centriole. It will continue as axoneme and it forms the axis of the tail. So this portion is called as the tail. So the axis of this tail is nothing but axoneme, or the continuation of axial filament. So this is regarding the structure of a human sperm. So the tail, this is the last part. It is again differentiated into main piece and the end piece. So this is a main piece and this is the end piece of human sperm, the tail of human sperm. The next very important component, that is a semen. What is mean by semen? So semen is nothing but seminal fluid with sperms. So semen is different, sperms are different. Sperms are found in the seminal fluid, together known as semen. Then where this seminal fluid is produced and where these sperms are produced. Sperms are produced in the testes. That means in the seminiferous tubules. Seminal fluid are produced by the glands, mainly the seminal vesicle and the prostate gland. And even one more gland is also there, that is the bulbourethral gland. So semen is the seminal plasma along with the sperms constitute the semen. The seminal plasma, the seminal fluid or the seminal plasma is rich in fructose, calcium and certain enzymes. The detailed account on seminal fluid will be discussed later. The secretions of enzymes will be discussed in detail later. So the seminal plasma is rich in fructose, calcium and certain enzymes, and during the ejaculation, the healthy male individual releases nearly 200 to 300 millions of sperm. That means the male individual during single ejaculation releases nearly 2 to 3 ml of semen. 2 to 3 ml of semen. See, 1 ml of semen consists nearly 100 millions of sperms. 100 millions of sperm. That means if the male individual releases 2 to 3 ml of semen, that produces or that consists nearly 200 to 300 millions of sperm. If the male individual releases 5 ml of semen, that consists nearly 500 millions of sperm. See, in case of a male individual, for the normal fertility, at least 60% of the sperms must have normal shape and size. Just now we discussed the structure of the human sperm, the shape of the sperm, the size of the sperm. That should be normal. At least 60% of the sperms produced by the individual must have the normal shape and size. And out of which about 40% of them must show the vigorous motility, So only if the sperms have the normal shape and size, only if they are motile, then only such male individual becomes fertile. And these semen are transported mainly first the sperms followed by the semen. They are transported by the help of accessory ducts, and we discussed about the different accessory ducts of male reproductive system. First, the sperms produced inside the seminiferous tubules are transported into the retestis, followed by the vasa efferentia. Then they are transported into the epididymis. So in the epididymis, the temporary storage of sperms takes place, the maturation takes place, and from epididymis, vas deferens, through the vas deferens, the sperms produced in the testis will come into the abdominal cavity, pelvic region. and then from vas deferens into the seminal, it will be transported through the ejaculatory duct. But before that, the seminal vesicle secretes a fluid that is called a seminal fluid. Even the prostate gland also contributes for the development or the production of seminal fluid. The secretions of these seminal vesicle as well as prostate gland called a seminal fluid, plus the sperms transported through these epididymis vas deferens together constitute semen. At the same time, the secretions of epididymis, secretions of vas deferens, secretions of seminal vesicle and the prostate are essential for the maturation and motility of the sperms. So remember, the transportation of sperm takes place through the epididymis vas deferens, followed by the ejaculatory duct and the urethra. At the same time, the secretions of seminal vesicle, prostate gland, bulbourethral gland, called as seminal fluid, along with the sperm forms the semen. At the same time, the secretions of epididymis vas deferens, seminal vesicle, prostate gland required for the maturation and motility of these sperms. Then, the functions of sperm, the functions of male accessory ducts, the functions of the male accessory ducts or the sex accessory ducts and glands are maintained by the testicular hormones. Maintained by the testicular hormones are also called as androgens. So androgens are nothing but the testicular hormones or the male sex hormones. These are nothing but the male sex hormones. So all the functioning of male sex accessory ducts as well as the glands are maintained by these hormones. Now, some additional points mainly related to the secretions of various glands of male reproductive system. The secretion of a seminal vesicle contains fructose, citric acid, ionositol, and prostaglandins. So this is mainly the secretions of seminal vesicle. I repeat, the secretion of seminal vesicle contains fructose. It is provided as a nutrition to the sperms, and citric acid, ionositol, and prostaglandins. The prostatic fluid, that means the secretion of prostate gland, provides characteristic odor or smell to the seminal fluid. So the secretion of prostate gland, that is the prostatic fluid, provides a characteristic odor or smell to the seminal fluid. See, the prostate gland secretes citrate ions, calcium, phosphate ions, and fibrinolysin. See, these are the secretions of prostate gland. I repeat, prostate gland secretes citrate ions, calcium, phosphate ions, and fibrinolysin. So these are the secretions. These are the secretions of prostate gland. So in our textbook, only the fructose, calcium, and certain enzymes are mentioned. So the semen has a slightly alkaline pH, that is around 7.2 to 7.77. 7 is neutral, more than 7, 7.2 to 7.7. So this is slightly alkaline pH. So it is required for the survival of sperms. Normally, you know, the urethra is a common passage for the sperms as well as the semen or the sperms. Just imagine, just before the ejaculation, if there is a urination, the releasing of discharging of urine, the urethra becomes acidic. If the sperms are exposed to such acidic condition, the sperms may die. So neutralize it, there is an alkaline pH. The prostatic secretions gives semen a milky appearance. Normally, the semen produced or ejaculated by the male individual, that is milky in appearance, whitish in color. So that milky appearance is because of the secretion of prostatic gland or the prostate gland, whereas fluids from seminal vesicle and bulbourethral glands give it a sticky consistency. So that is sticky in nature, milky in appearance. The sticky nature is mainly because of the secretion of seminal vesicle and bulbourethral glands, and the milky appearance is because of the secretion of prostate gland. The next type of gametogenesis, that is oogenesis. So oogenesis is found in the female individual. As I mentioned earlier, there is a difference in the beginning of gametogenesis in males and females. In females, the oogenesis begins. The oogenesis begins at the time of embryonic stage itself. So during the development of female child itself, the oogenesis begins. So how it occurs? See, here is the diagrammatic representation of oogenesis. This is a very simplified diagrammatic representation of oogenesis. So let me explain the steps involved in it. The process of formation of mature female gamete in the ovary is called as oogenesis, and it is initiated during the embryonic development stage itself. That means a couple of million gamete mother cells called as oogonia are formed within each fetal ovary. At the time of embryonic development itself, in the fetal ovary, a couple of million gamete mother cells or oogonia will be there. That means no more oogonia are formed and added after birth. So the time of the birth itself, the newborn female child, the newborn female baby, its ovary consists a couple of million gamete mother cells or the oogonia. Remember, no more oogonia are formed after the birth. That means the phase of multiplication is already completed before the birth of that child. The phase of multiplication, the mitotic division that occurs even before the birth of the female child. So in the embryonic stage itself, the phase of multiplication will be completed. So after the birth of that female child, what happens? The oogonia, that is a gamete mother cell, they will start to divide meiotically. Even before attaining the sexual maturity, the meiotic division begins. That means, actually here also the just like the spermatogonia, oogonia are formed in case of females. The oogonia undergoes a phase of maturation. Phase of growth, that is increase in the size. The enlargement of the oogonia takes place, that leads to the formation of primary oocyte. Now this primary oocyte enters into meiosis 1. That will not complete the meiosis 1. They will just enter into the meiosis 1, and such cells, primary oocyte, they will enter into the meiosis 1 and they will be arrested in prophase 1 stage. Just remember, before entering in or before attaining the sexual maturity or puberty, the oogonia cell undergoes maturation, that leads to the undergoes cell enlargement or growth, leads to the formation of primary oocyte, and such primary oocyte enters into meiosis 1 and are arrested in prophase 1. So they are temporarily arrested in prophase 1. So these structures are at present, before the completion of meiosis 1, they are considered as primary oocyte. Now, these primary oocyte at the time of maturity, all these events takes place. That means just before the attaining puberty, the primary oocyte develops or surrounded by a layer of granulosa cells called as the primary follicle. That leads to the formation of structure called as primary follicle. Remember, before the attaining of sexual maturity, around the primary oocyte, around the primary oocyte, a layer of granulosa cells are developed. At this stage, the primary oocyte surrounded with the granulosa cells, a layer of granulosa cells, is called as primary follicle. So primary follicle internally consists primary oocyte arrested in prophase 1 and externally surrounded by a layer of granulosa. So that structure is called as a primary follicle. Now, a large number of these primary follicle, that means primary oocyte, undergoes degeneration during birth to puberty. So before puberty, all these events takes place. That is the development of primary follicle. But at the same time, from birth to puberty, a large number of these primary oocytes or oogonia or the primary follicle undergoes degeneration. That means all the couple of million gamete mother cells or oogonia will not develop as functional primary follicle. They will not continue as a functional primary follicle. A large number of primary follicles will be degenerated during the phase of birth to puberty. So the time of puberty, they consist a limited number of primary follicles. So at puberty, only 60,000 to 80,000 primary follicles are left at each ovary. So this is a very important point. At puberty, only 60,000 to 80,000 primary follicles are left in each ovary. One more thing, all these 60,000 to 80,000 primary follicles will not develop as eggs because the duration of reproductive phase is limited in female individuals. The gametogenesis process can occur, or the production of gametes can occur only during the reproductive phase. The duration of reproductive phase in female individual is of short period. So normally at the age of 12 or 13, at the age of 11 to 13, the female individual attains puberty. So at the age of 11 to 13, the female individual attains puberty. So at that particular age, she will start to produce the eggs. And the process of gamete production will continue at the age of, till the age of 45 to 50, normally 45. After that, there is no production of gametes. So just imagine 11 to 45. This is the age gap during which the gametes are produced in female individual. And you know the time required for the production of one egg, that is 28 days. Consider it as 1 month. So for a month, only one egg is produced. So there is no time for the conversion of all these primary follicles into eggs in female individuals. And here is a diagrammatic representation. Let us see the steps involved in gametogenesis. Before that, see the more layers of granulosa cells. You know, at the time of puberty, the ovary, each ovary consists 60,000 to 80,000 primary follicle. Primary follicles are nothing but primary oocyte surrounded by a layer of granulosa. Now, more layers of granulosa cells are developed around the primary follicle. More layer of granulosa cells as well as new thicker layers, protective layers are also developed around the primary follicle. So that leads to the development of secondary follicle. Primary follicle will develop as secondary follicle. I will show the diagram. So this, if it is the ovary, the development of follicle, ovarian follicle takes place only in the margins. So the time of puberty, just imagine this is a primary oocyte, and that primary oocyte is arrested in prophase 1, and this primary oocyte is surrounded by granulosa cells. So primary oocyte with the granulosa cells. So this is called as primary follicle. This is a primary follicle. So many number of nearly 60 to 80,000 such primary follicles will be there in the ovary. So I will show only one primary follicle here. In the next step, what happens? Few more. Just imagine this single primary follicle. So this is a primary follicle. This is actually the primary oocyte surrounded by granulosa cells. So these are the granulosa cells, single layered granulosa cells. So now what happens? Few more granulosa cells are developed. Okay. So now at this stage, some protective layers called as thicker layers are also developed. So now it is called as a secondary follicle. So this is a secondary follicle. Again, it further undergoes maturation. It further develops as the matured structure. Again, this is a primary follicle. Remember, during this maturation process, during this stage, this primary oocyte will continue the meiotic division. Actually, at the time of puberty, all the primary follicle consisting primary oocytes, such primary oocytes are arrested in prophase 1. But when there is a development of ovarian follicle, that means the development of primary follicle as secondary follicle, tertiary follicle, Graafian follicle, they will continue the meiosis. They will continue the meiosis. Prophase 1 followed by metaphase 1, anaphase 1. So all such events takes place simultaneously. So now few more granulosa cells are formed. Few more granulosa cells are formed. So few more granulosa cells along with the formation of an internal cavity also takes place. There will be the formation of an internal cavity also. So like this, the tertiary follicle is developed. So at this stage, at the time of a tertiary follicle development, what happens? There will be the formation of a small space within, there is a formation of a fluid-filled cavity within the ovarian follicle. So now this is a primary follicle, primary oocyte, actually that has continued its meiosis 1. So like this, the granulosa cells. So here is the formation of cavity. You can see the cavity now. So that cavity is filled with, fluid. That cavity is filled with the fluid. So this is the cavity called antrum. And now it completes its first meiotic division. At this stage, this primary oocyte, which was arrested at prophase 1 before puberty, they will continue the meiosis 1 and they will complete the meiosis 1. At this stage, this is called as secondary oocyte. At this stage, it is called as secondary oocyte. So it completes its first meiotic division, unequal division. But remember, this meiosis 1 is the unequal division. Unequal division means the primary oocyte, it undergoes meiosis 1, leads the formation of one large size cell, one small size cell. So it produces a large haploid secondary oocyte. The large size cell is called as the secondary oocyte. So this is the secondary oocyte. And a small cell is called as polar body. So this is also haploid and this is called as a polar body, first polar body. So this is a polar body. So this is non-functional, but this one is the functional. Okay. Now at this stage, the ovary consists of fully matured ovarian follicle. That fully matured ovarian follicle is called as Graafian follicle. At one particular day, that is on 14th day of menstrual cycle, the ovary ruptures and that releases ovum into the oviduct or the fallopian tube. So the Graafian follicle ruptures, that releases the ovum. That process is called as ovulation. That process is called as ovulation. Now, at this stage, before the rupturing, the Graafian follicle consisting the secondary oocyte. That secondary oocyte is covered by a non-cellular covering that is called as a zona pellucida. So the rupturing of Graafian follicle releases the secondary oocyte by the process called as ovulation. So this is regarding the oogenesis. Now let me explain the schematic representation. In schematic representation, the process of development of primary follicle, secondary follicle, tertiary follicle, etc. are not given. Just only the division process, the process of gamete formation is mentioned here. So this is oogonia. The phase of multiplication that occurs during the embryonic stage itself, So the time of childbirth, the female child consists of a large number of, a couple of millions of oogonia will be there. So after the birth, there is no further addition of oogonia. So among these, these oogonia then undergoes cell enlargement, nothing but the phase of growth. So that leads to the formation of a primary oocyte. Now, after the birth till the puberty, this primary oocyte undergoes a further development. So at this stage, the primary oocyte, around the primary oocyte, there will be the development of a layer of granulosa cells. So now it is called as a primary follicle, but it is not mentioned in this diagrammatic representation. But actually during this period what happens? The primary oocyte is arrested in the prophase 1. But after maturity, after puberty, this primary oocyte, which is found in the form of primary follicle, undergoes a further development. There is nothing but the maturation phase, that is the completion of meiosis, during which meiosis 1 of primary oocyte produces secondary oocyte and polar body. So in case of spermatogenesis, both the cells are called as secondary spermatocytes. But here, primary oocyte produces single secondary oocyte and a small sized first polar body. So this polar body is actually non-functional. The fate of this first polar body is not clearly understood. But in this diagram, its meiotic division is shown here. But the very important cell here is secondary oocyte. What about the secondary oocyte? The secondary oocyte then completes the second meiosis, and the second meiosis leads to the formation of oocyte or ovum. But as I mentioned, as I told earlier, in case of human beings, during the ovulation, in case of human beings, during the ovulation, there is a releasing of secondary oocyte. Secondary oocyte means it is yet to complete. It is yet to complete second meiosis. So the egg second meiosis is yet to complete. So the egg produced during this ovulation is not completely developed. That means when it will complete the second meiosis. Remember, the second meiosis takes place only after the sperm penetration. Only after the sperm penetration. So after the entry of sperm into this secondary oocyte, it will continue the second meiosis and it produces oocyte. It produces oocyte and polar body. The polar body will be eliminated out, and oocyte, it now undergoes. Now this oocyte undergoes fertilization that leads to the formation of zygote. So this is a special phenomenon found in female individuals in human beings. So here is the diagrammatic representation of mammalian egg or ovum. The mammalian egg ovum. So just see the important structural components. You know, during the ovulation, it releases, the ovary releases secondary oocyte. The secondary oocyte consists of different structural components. The outermost layer that is called as corona radiata. So this is a corona radiata. So this is a cellular covering protective layer called as corona radiata, and is followed by there is a non-cellular layer. This non-cellular layer is called as, this is a corona radiata. This non-cellular protective layer is called as zona pellucida. The zona pellucida is followed by white line membrane. Normally it is formed at the time of fertilization. So it prevents the further entry of sperm. White line membrane, fertilization membrane. And then there is a plasma membrane. So there is a plasma membrane. So this is the membrane of egg. Plasma membrane. It is also called as oolemma. And then it is a cytoplasm. There is a cytoplasm within the egg. The cytoplasm of egg is called as ooplasm. It is also called as vitellus, or it is also called as yolk. Ooplasm, vitellus, or yolk, one and the same. And then it consists the nucleus. There is a germinal vesicle. So this is a germinal vesicle, and within which there is a nucleoid that is called as a germinal spot. So this is the structure of mammalian egg. Now let us see some additional points related to the egg, the female gamete, egg. There are important points. That is, based on the amount of yolk, we can classify the eggs into different types. So this is related to egg. The classification of egg of animals into different types based on the amount of yolk. In that, first one is alecythal egg. Alecythal egg means here the yolk is absent. Yolk is absent. The egg without the yolk, is called as alecythal. Remember, human egg is an example for alecythal egg. The another type, microlecythal. Microlecythal means the yolk, the amount of yolk is very little. So that's why it is called as microlecythal. Most commonly, the starfish egg is an example for microlecythal egg. The next one is mesolecythal. Meso means the middle or moderate amount of yolk is found. The mesolecythal eggs are commonly found in frogs. The next one is megalecythal. Megalecythal egg is also called as macrolecythal egg. And here the large amount of yolk is found. And most commonly the reptiles and birds produces megalecythal or macrolecythal eggs. So this is mainly based on the another classification based on the distribution of yolk. So the first one classification based on the amount of yolk. Now based on the distribution of yolk within the egg. If the yolk is uniformly distributed within the egg, such type of eggs are called as isolecythal. Example, Brachyostoma as well as Herdmania. The next one is telolecythal. That means the yolk is concentrated towards one pole. That pole is called as vegetal pole. The opposite pole is called as animal pole. So vegetal pole means the region of the egg towards the yolk is concentrated. Normally the amphibians produces a telolecythal egg. Next one is a centrolecythal. In case of centrolecythal egg, the yolk is present at the center, and most commonly the insects produces a centrolecythal eggs. So this is regarding the very important points related to the structure of male reproductive system as well as the female reproductive system, including along with the NCERT topics, some additional points are also given. So just go through it. So in the next video, in the another video, I will discuss some important MCQs on the chapter human reproduction. Thank you.